pass them to Spenser at the end of his presentation. So with that, I will introduce today's speakers and let him get on with the presentation. So it's my pleasure to introduce Spenser. So Spenser Widin is a biologist with the U.S. Geological Survey's Great Lakes Science Center. They are focused on field and laboratory development of novel treatment strategies for managing the invasive Phragmites australis and other non-native wetland plants. Currently, he's working with collaborators to improve the efficacy of microbial-based approach to Phragmites management, which I'm sure he'll talk about more in this presentation. So, Spenser, you can share your slides and take it away.
There we go. Are you seeing that okay there, Teresa?
Yep, that looks good to me.
Perfect. All right. Well, thank you for that introduction, Teresa, and thank you everyone for being here today to learn more about the work we're doing to help support land managers in their efforts against controlling the invasive Phragmites australis, and particularly some of the work we're doing with microbial-based approaches to that control. And so before we jump right into the science of this, just like to take a moment here to acknowledge the Great Lakes Restoration Initiative, which has provided the funding for this collaborative effort between our team here at the USGS, led by Dr. Kurt Kowalski, as well as the team at Rutgers University, led by Dr. James White and Catherine Kingsley over there. So this is me here presenting today, but it's definitely a group effort between many different people involved to make this happen. And so, while most people here are joining because they're probably pretty familiar with Phragmites, well, this talk really wouldn't be complete with me at least setting the stage for the work that we do. And so, not to be confused with its native counterpart, Phragmites australis subspecies americanus, the work we're doing is to help with the control efforts for its introduced or invasive counterpart, Phragmites australis subspecies australis. And so, as most of you are aware, it's a perennial wetland grass species that has been introduced into North America. And since its introduction here has spread throughout pretty much all of the Great Lakes region and really North America at large. And since its introduction and because of its growth patterns, it can form these really dense, tall, nearly monotypic stands that can reach, upwards of 5 meters tall, that can come with a whole host of different negative impacts, not just for the environment through reductions in biodiversity and degradation of wildlife habitat, but also impacts for society through restricted access to recreational opportunities, as well as some significant economic impacts through things like reduction in home values for homes that occur along these shorelines that have been invaded by Phragmites, or Phragmites invasions that have occurred along critical infrastructure, making it harder to access that infrastructure and making it more costly to maintain and service these type of things. And so while this plant, we're mostly familiar with the above ground parts of this plant because it's a really prominent part of the landscape. It's one of the really challenging aspects of managing this actually has to do with what occurs below the ground, the things that we can't see. And so while there is a lot of above ground biomass that occurs in the areas where Phragmites has invaded, there's oftentimes just as much, if not more, below ground biomass that occurs, largely in the form of these structures called rhizomes, which you can see me holding one here in the center image here. These rhizomes are essentially like modified root structures that serve a number of different functions for the plant. Two main ones being helping the plant spread to new areas and storing resources for that plant to be able to regrow in subsequent seasons or following disturbance events, sometimes our own disturbance events like some of our management actions. And so in terms of spread, these rhizomes can actually grow quite rapidly within a season. And we've seen them grow oftentimes meters within just a span of a few months. And so it's kind of hard to see in this bottom left-hand image here, but this is from several years ago, a greenhouse experiment we did. And I'm holding a rhizome here with one of our student interns, Annie, there, stretching out this long rhizome that grew in only about 4 or 5 months time, I think from about May to September. So they can grow really, really rapidly, spread once they've invaded an area, and form these really dense network of rhizomes under the surface that can store quite a lot of resources in the form of carbohydrates. Sometimes we've seen around 30 to 40% of that rhizome can have resources for storage for future growth and things like that. So really challenging problem to deal with because of this really extensive underground network that can spread and produce new growth in the future. And also they have a really high seed output. Like you can see in the top right image here, they produce these kind of fluffy seed heads that are wind dispersed at the end of the season, and those seeds can travel quite some distance and introduce new invasion populations across the landscape. And so all of these things make Phragmites a really difficult problem to manage. And there are some traditional approaches that I'm sure most of you are familiar with, like prescribed burning of that above ground biomass, applying herbicides, using cut to drown or cutting the plants underwater to starve them of oxygen, and mechanical removal of those above ground biomass in kind of drier areas and things. And while these management treatments can be effective, they often require successive years or even within the same season, repeated treatments for them to really have a strong impact. Oftentimes they're also not species specific, and they can even have some limited or restricted uses in certain areas, like herbicides use in aquatic environments up in Canada. It can be quite restricted. And so all of these things lead to, yes, these treatment methods can be effective, but they're quite labor intensive and quite costly to implement. And so the management community has articulated some needs for additional tools that they can use to increase efficiencies and efficacy of treatments for this problem. And so that's kind of where our research program comes in. We're trying to address that need that managers have to be able to control this kind of really difficult plant across the landscape. And so here at the USGS,
sorry. Yep. I don't want to interrupt your presentation. There's like I think your computer is trying to connect to like the global protect.
Yes, and it's there it goes.
You got it.
For the interruption.
Thank you.
I appreciate that. Yeah, so as I was saying, we're kind of stepping in to assist managers and develop some of these additional tools for them to be able to combat this difficult invasive plant. And this kind of takes two different approaches within our lab. The first one being a genetic approach, which is not the focus of the talk today, but I do want to highlight some of that work that's being done with some of our collaborators in the Army Corps of Engineers, and there we go, through the Army Corps of Engineers Engineering Research and Development Center down in Vicksburg, Mississippi. And this work down there is largely led by Dr. Ping Gong, where they're looking at some gene silencing or RNA interference techniques down there. And so I'm not going to focus on this during this talk, but I do want to point to just these two resources here where you can learn more about this really promising new technology. There was a past webinar that Dr. Gong gave for the Great Lakes Phragmites Collaborative that you can find on their website, as well as a very recent publication that came out earlier this year on some of the advancements in this technology that they've put forth, moving forward, moving closer towards some field testing of this technology, hopefully pretty soon. And so check those resources out if you're interested in learning more about that collaborative effort we got going on. But for today's talk, we're going to be focusing on this microbial approach to Phragmites management. And so this too is another collaborative effort with Rutgers University, again led by Dr. James White, in which we're trying to utilize what we've learned about the plant microbe interactions to leverage that as a way to reduce the invasiveness and ultimately induce plant death for Phragmites. And so if we look a little bit closer at what this microbial relationship entails, just like us humans and animals, plants also have a microbiome associated with them in the form of fungi and bacteria that occurs throughout all of the stages of their life, from a seed through development into a seedling, into a mature plant, and then kind of repeating that cycle into a senescing plant producing seeds again. And while these relationships between the fungi and the bacteria and the plants can take a number of different flavors, from beneficial to neutral to negative interactions, the one that I kind of want to focus on here is the beneficial relationship that happens between plants and microbes, particularly in the soil or the rhizosphere. There's this thing that occurs that work from Dr. Kirk Kowalski and James White and many others have termed the rhizophagy cycle or the rhizophagy cycle. And essentially what happens is the plants and the microbes are interacting in this beneficial way where the plants can release molecules from their root system that essentially is food for the microbes. And in exchange for that food, the microbes can make some of those soil nutrients available to the plant. And so there's been some studies to show just the importance that these microbes play in the growth of plants, and Phragmites here in particular, in terms of growing with or without microbes. So a study done by Ernst et al. In 2003 showed that when you grow some Phragmites plants in soil, shown here on the left, that's been sterilized or stripped of their microbes, it has really negative impacts on the growth and the health of the plant, whereas Phragmites grown in some soil that has been intentionally inoculated with some microbes shows really significant improvements in the growth and the health of the plant. And so these are well-documented interactions, and we're essentially trying to utilize that information and sort of dial the levers in a way to flip that interaction on its head and take it from one that often benefits the plant to one that kind of has a negative influence on the plant. And so that seems like kind of a big task. And so where do you start with that approach, right? And so to address that question, a team of individuals, including people from here at the USGS, academic organizations, other federal organizations, as well as the Great Lakes Commission, formed the Phragmites Symbiosis Collaborative to set out a series of conceptual strategies to be able to ultimately work towards this goal of a microbial-based approach for Phragmites management. And so in 2015, they published a paper here in Frontiers in Microbiology that laid out this strategy, essentially starting from identifying those microbes that are really influential in Phragmites, to then moving on to identifying some of those particular relationships that could be influenced for control, to ultimately testing the feasibility in the field of some of these mitigation strategies influencing this microbiome, which is the focus of the talk today, is some of the testing that we've been doing in the field over the last several years of some products that showed some really strong promise within a laboratory and a greenhouse type setting. And so this has taken the form of what we're calling a bioherbicide product because it's influencing the biology associated with the plant. And you can see here on the left-hand image the components of that bioherbicide formula being citric oil, which comes from cold-pressed orange peels, arginine, which is an amino acid that all living organisms on Earth require, sucrose, which is a fancy way of saying sugar, glycerol, which is another naturally occurring kind of sugar-based molecule, and then all of these things are dissolved in water. And so we've been working on this for a while, like I said, in greenhouse, laboratory, and now the field. And so there is some patents out there for both the US, Canada, as well as the international patents for this product. And like I said, this has been tested in the lab in the field with really pretty good success initially early on. And I can kind of take you through here why it is that we include all of these naturally occurring, readily biodegradable, relatively safe for human kind of components here. And so first off, we have the citric oil, which essentially is like a degreasing agent that when applied to the surface of the leaf in conjunction with the glycerol, which acts as a wetting agent to kind of disperse this across the surface of the leaf, acts to strip away that waxy layer that occurs on the outside of plant tissues that is there to protect the plant from things getting in as well as water loss. So we add the citric oil as a way to kind of remove that barrier for the addition then of arginine, which through prior testing with Dr. White and others at Rutgers have shown that when you supply extra arginine to microbes associated with Phragmites, it can kind of cause this pathway to happen that causes the microbes to release excessive amounts of ethylene, which is essentially a plant hormone that can lead to stress and senescence and ultimately cell death of the plant. And so in small doses, the arginine kind of works in benefit with the plant. The plant normally has control and can release that to the microbes. But by us supplementing that, we can kind of induce this pathway that leads to this excess of ethylene production, causing stress and ultimately, hopefully leading to cell death of the plant. And then the sucrose, or the sugar there, is essentially just food for these microbes to keep them happy, keep them fed, keep them working on this arginine as kind of this feedback of cell death and ultimately, hopefully, leading to reducing the invasiveness of this plant. So this is the product that we've been testing both in the laboratory, but and now recently in the field. And so I'm going to be focusing on the last 3 years of our field testing of this product. And so, like I said, in the laboratory and in the greenhouse setting, we've had some early initial successes with showing really detrimental impacts to the growth and development of Phragmites. And so we wanted to take that to the field to then test the efficacy in a field setting, because the greenhouse, nice controlled environments, but the field, as we know, has a lot of variability there. And so we wanted to do some initial tests to see, is this product impact's going to translate into a field setting. And so on top of that, we tried to address some initial questions regarding application strategies of this product. So timing, whether early in the season, late in the season, or frequency of application, essentially how many times throughout the season do we need to apply this product? And then also looking at the method of application in just a strict foliar application, so to the above ground parts of the plant, or in the soil application, getting closer to that rhizosphere, closer to the microbes and the roots and the rhizomes of this plant. And then ultimately looking at adjusting the formulation, adding or removing certain ingredients to kind of dial in the optimal formulation for impact, and then looking at whether our product has systemic versus some contact effects. And so over the last 3 or so years, we've been testing this in the field in a series of experiments that more or less follow the same series of events. When we locate a new Phragmites patch, we'll go out there early in the spring and clear it of all the standing dead stems, essentially down to the soil, where we then can stake out these 1 meter by 1 meter experimental plots. Where we then take our root spade here and sever around the exterior of all of those plots, essentially trying to cut off the rhizomes from the surrounding patch. So isolating that patch, because the surrounding patch can share resources with the treated patch. And so we're trying to limit that the best we can. We then apply our treatments at routine intervals throughout the field season, and then perform weekly monitoring of plants' growth and stress, and kind of repeat this treatment monitoring cycle. And so we've been working all over Southeast Michigan with this for now the 3 years. This will be the 4th year this coming summer here at numerous different sites, just 3 of which I'm going to touch on in this talk, because 3 years of work's a lot to fit into a 40-minute talk here. And so the very first site that we went to is called North Hydro Park. It's an Ypsilanti Township property that is located in between Belleville and Ford Lake along the Huron River here. And we've been at this site now for 3 years performing this work. Our initial tests at this site, we're looking at that question of application timing. And we kind of had 2 different blocks of treatment, which I'll get into more. Then in the second year of testing, we added a different site called Crossman's Marsh, which is a Wayne County Parks property, located kind of south of the Detroit airport there on the map. And we started this in 2023 and have done 2 years of testing at this site, where we looked then at that question of application the timing at this one. And then finally, last year, we added a 3rd site called the Crystal Waters State Game Area, which is a Michigan Department of Natural Resources site that used to be a former sand and gravel mine that now has quite a bit of Phragmites on there. And at this site is where we started to look at methods of application and introducing some soil applications of this. So first, I'm going to start with our North Hydro Park site, where we looked at some initial foliar application tests to look at this question of application frequency. And so at this site, we had 30 total plots that were in 2 discrete patches. You can kind of see our plots here from this aerial image, one patch up here, and then another patch kind of lower down this hill. And here we looked at a really intensive application of this in 2-week intervals versus a less intensive application in a 4-week interval treatment of this. And so if we first kind of look at some visual impacts of our Phragmites treatments, kind of on this like 4-week treatment cycle, pretty quickly within a week, and honestly within a couple days of applying our product, we see some pretty stark impacts on the plants through discoloration of the leaf as that cuticle gets stripped off, a lot of drying and desiccation and dying of tissues as water loss occurs, and kind of curling of leaves. Then within about 2 weeks' time, we start to see evidence of a microbial bloom happening on the surface of the tissues of this through fungus and mold growth on the exterior. And then within about 3 to 4 weeks' time, we do see some recovery of these plants, but ultimately it's with a lot of signs of stress in terms of deformities of their growth here, which you can see, and kind of this characteristic stress response of putting out a whole bunch of shoots from one of those nodes. And so from here, we can then look at our routine monitoring data that we collected and see how these visual effects we were seeing kind of compound throughout the season. And so starting here in our very first initial test in this top left figure here, in year 1, this figure here is showing Phragmites percent cover, which essentially is a visual estimate of the leafiness, if you will, of these plants, how big and how bushy they're getting. So the y-axis is looking at percent cover, and then the x-axis here we have time points throughout the sampling season. And from this, you can notice that pretty much across all of our treatments, control and the bioherbicide applicated treatments, you can see a steady increase in Phragmites cover through time as plants continue to grow. And that's because this metric is really encapsulating all of the leaves on the plant, whether they're alive or whether they're dead. And so plants were, like I said, recovering and producing new leaves, and the impacted leaves were kind of holding on still until later in the season is when we started to see a really rapid decline in cover compared to controls. But when looking at another aspect of our routine monitoring in terms of mortality of those above ground stems, we can see here a really strong impact of our bioherbicides. So in this figure, we're looking here on the left side. On the y-axis is the average dead stem percent. So the percentage of the total stems within that plot that are experiencing death due to this bioherbicide product, and then again throughout the sampling season here. And you can see across both of our treatments that we really get a significant impact in terms of mortality of those above ground stems, and you can see that visually here with these photos from the end of the season where those plants, lost most of their leaves, those stems are looking really dead, and those sort of things. And so these impacts that we saw in year 1 translate it to year 2 and year 3. And so if we look at our results from the 3rd year of this test, here we saw similar impacts in year 1. And after year 1, while our above ground stuff did show some differences between 2 and 4 week, when we looked at our below ground data, which I'll get to next, those differences were small enough that we thought that maybe the extra effort involved with a 2-week treatment wouldn't be warranted. And so moving forward in years 2 and 3, we actually only just applied all of the treatments in a 4-week interval. And so that's why you kind of see 2 and 4 week plots kind of tracking one another in year 3 here. So similar trend of increasing Phragmites cover through time. But again, when we looked at the mortality of those stems, we saw a pretty significant increase in mortality of plots that received this bioherbicide treatment. And so if we then look at how this above ground impacts translated to our below ground effects about all those rhizomes I talked about in the resource storage at the beginning, we are able to sample rhizomes from our plots at the end of the season and analyze how many carbohydrates are in them, starches and sugars for the plant, the energy molecules of this plant that can support regrowth. And so here in this figure, we have on the y-axis or the vertical axis, we have our percent total carbohydrates. So of the rhizome tissue we collect, what proportion of that or what percent of that is these resource molecules, these carbohydrates that are stored. And from this, you can see some really significant impacts in the storage capacity of plants that were treated with our bioherbicides. So pretty significant reductions in how much storage they were able to send down to their roots throughout the year. And in year 3,
Spenser, I'm so sorry to interrupt again. The Global Protect is back. It really wants you to sign in.
Disconnect. Let's see if that helps. Thank you, Teresa, and apologies
You're welcome
for that. So again, looking at year 3, after now 3 years of applying this bioherbicide product, we see similar reductions in the below ground resources of this plant, but those reductions have a lot more variability in them. And that we have a couple explanations for that, but we're largely attributing that to some site level differences that occur. So here we are looking back at the aerial of this site that we worked at, North Hydro Park, where in our experiment, we grouped these treatments into 2 different patches of Phragmites. Patch 1, which is up on a hill and kind of flat, and patch 2, which is down on a hill and more of a slope. And so again, here is that carbohydrate figure, the resource storage figure we looked at, but now taking a little bit more finer look at it and separating out results based on those 2 patches. So on the left of this is the upper patch, and in the right, this is patch 2 on the lower side of this site here. And from this, you can see that there is pretty significant reductions in carbohydrates across the whole site, regardless of where those plots were located here. Whereas in year 3, here, same figure, but now looking in year 3 or 2024 last year, it was really in patch 1, that upper patch, where we saw that strong reduction in below ground reserves. And that upper patch, we saw some reduction, but there was a lot more variability in there. And we have some potential hypotheses as to why that happened. And there are some things there for us to poke at in the future and possibly investigate this coming field season. And so just kind of in summary, one more slide here. This is just some visual impacts to kind of highlight the differences we saw between that upper patch 1 and that lower patch 2 compared to our controls. And so you can see really that below ground data we saw kind of translated to some of those above ground visual impacts as well, where our upper patch there, patch 1, much harder hit by our bioherbicide product than in patch 2. And so what we kind of took away and learned from this first series of treatments, looking at the frequency of application, is that year over year, we had pretty significant impacts on that above ground parts of the plant through reductions in cover, increased mortality of stems, as well as increased stress and reductions in their ability to kind of grow. We did see pretty significant reductions in the below ground resource ability, storage ability of these plants through some of this, impact on the above ground, not being able to translocate some of those resources below. But we did see some site level variability start to emerge between our 2 patches. And so again, there's some things there for us to look at going forward. And additionally, we also, after that first year, in terms of our below ground stuff, saw a small difference between 2-week and 4-week, but not enough, may not have been enough for us to warrant that extra labor and materials for this really intensive kind of 2-week treatment method. So moving forward, all of our treatments at our new sites and new experiments kind of followed this 4-week pattern. And so after these initial tests of frequency, we kind of turned to now looking at the timing of application of this and kind of aligning it more with maybe how some more traditional treatment approaches occur, like later in the season, like things like glyphosate treatments. And so for this, we added a new site, again here, called Crosswinds Marsh, which is a Wayne County Parks site. We added this site in 2023 and have worked there for 2 years now. Here we had a little bit larger of an experiment. We had 40 total plots that we then looked at adding some additional treatments to, either combining with some traditional approaches like cutting the plants early in the season and then applying subsequent foliar applications of our treatment, or really looking at the timing of application of treatment. So either applying treatments early in the growing season and then subsequently afterwards, or applying a late season treatment, so late summer, fall treatments, kind of like you would do with glyphosate, to take advantage of the senescence of the plant, and when they're dying, they're moving all of the resources that they've made through photosynthesis down into the roots, and hopefully taking advantage of that and getting a systemic impact of our plant. And so again, if we turn to similar set of monitoring results from this test of the application timing, here we can see again a figure showing the Phragmites cover through time on the y-axis, and then the date here on the x-axis. The vertical lines here indicating when treatment events happen. So in June is when we applied either our first cut or our early treatments, and then sprayed those same plots again in July. And then August is when our late treatment started. So similar again to when glyphosate and things would happen. And from this, what I want to highlight here is that in terms of the cover of the plant, where we saw the most impact was our early treatment, so kind of a more intensive treatment strategy, as well as our cutting treatment, because essentially any new growth that came up at the beginning of the season, we cut it back down to the soil and then continued applying treatments. Whereas our late treatment plants, we didn't see as much impact in terms of cover of the plants. And we monitored those plants at the beginning of the season, let them do their thing and grow, and then monitored them once more right before treatment. And at that point, we did see some reductions in cover, but not as much of an impact as we saw in our cut or our late treatments. However, when looking at the mortality effect of this, so again, here looking at the average proportion of stems in that plot that we observed to be dead, here we did see significant impact in terms of that late treatment, so a little bit less intensive of a treatment here. And by the end of the season, across all of our treatments, we had pretty similar impacts in terms of mortality of these stems. And so this is the data. But you guys are probably wondering, what does this actually look like, right? What do these plants look like when all of this has been done? And so here's some representative images of plots within each of these 4 treatments. So control on the left, towards the end of the growing season here in September, looking happy, healthy, green, nice bushy frag like you would expect, and then followed by our cutting treatment here. And here you can see, a reduction in the height of these plants, a reduction in cover, and a lot of brown, desiccated, and dead tissues there, as well as in the early treatments, similar types of impacts as to our cut treatments, and then late ones here, quite a bit difference in terms of the visual impacts of the above ground stuff, similar in cover to the control plants, but our treatments are having quite a big impact on the survivability of those stems, as you can see here with a lot of dead and brown tissues on those. And so those above ground impacts, just like we saw in years prior, translated to some below ground effects through similar reductions in below ground reserves. So again, here's that same kind of carbohydrate figure that we've been looking at. And in yellow, you can see the controls with a relatively high amount of that resource storage. And then all of our treatments here with pretty similar levels of low levels of carbohydrates or that storage capacity there. So this kind of showed us that depending on which time of the season we start applying this, we still seem to have similar impacts in terms of the ability of this plant to store resources throughout the growing season. And so just a summary here kind of what we learned from this second round of tests looking at this timing of our treatments, seasonality of our treatments. We saw a different magnitude of impact in terms of above ground effects, which you can see in these images here in terms of the early, which started in June treatments, and then the late, which started in July. But those above ground effects did not translate to the below ground effects in the same way, in that we had reductions in below ground reserves like consistently across all of our treatments. There was some variability, but we had a consistent reduction in those below ground resource reserves through time. But what we've noticed, and we've sampled rhizomes and kind of brought them back to our lab and tried to regrow them, we've noticed that these rhizomes do seem to remain viable through time, so they can support growth in the future, albeit with less resources, so possibly less growth in the future, but they do appear to be remaining viable. So to that initial question we were trying to look at of, is this more of a contact versus systemic effect? It appears that we're having more of a contact effect at this point through time. And so that led us to our next series of testing that we did, looking at the getting more directly to where the microbes are doing a lot of work with the plants in that rhizosphere, in the soil. And so here's where we added yet again another site looking at soil application of this. So applying our bioherbicide product to the soil to get in contact with the microbes, in contact with the rhizomes, to see if we could improve some of that systemic effect and some of the degradation effect to the rhizomes and the root material themselves. And so here is where Crystal Waters State Game Area comes into play. Again, this is a Michigan Department of Natural Resources site. And here at this site, it's a really large property. It used to be a former sand quarry, but it's actually quite a beautiful place now. But it does have a lot of Phragmites that occurs all along the shoreline on both sides, south and north, as well as a little bit up on the plateau here. And so at this site, we had a lot of opportunity for testing. It was a huge patch. We were able to go quite large here. We had 161 of those 1 meter square plots that we then spread across 3 discrete patches, either along the north side of the shore, the south side of the shore, or up on this plateau. So we kind of captured some different hydrologic regimes as well as soil types in our experiment here. And in this experiment, we looked at, like I said, soil application, but we also looked at how that compared to foliar applications and how that also compared to different levels of essentially feeding the microbes. So different levels of that sucrose or that sugar application. So we had 2 levels of that. We had a high level of sucrose, so essentially providing a lot of food for microbes. And we had a low level of sucrose concentration, so less food for them, that we then replicated across either strictly soil treatments of this, strictly foliar treatments, or a combination of soil and foliar treatments that we again applied in this kind of 4-week treatment strategy starting in about June through the end of September here. And so you're probably wondering, how did we accomplish these soil treatments? And here is a short video of me applying some of these soil treatments in the field, we used this backpack soil injector, it's called, which is typically used by arborists in tree management and things like that, but we've kind of adapted it for our work here. You can set the volume per injection on there, and we were able to then inject about 2 liters of our substance into the soil in kind of this grid pattern across the surface to hopefully get an even saturation throughout that experimental plot. And so again, following the same kind of sequence here I did with our other sites, turning to some of our monitoring data and what impacts this had on the above ground tissues. So I want you to get bogged down with just the extra amount of lines in here, simplify it for you a little. Again, this is Phragmites cover here. So on the y-axis, we have the average leafiness, if you will, of these plants. And then through time across our routine monitoring with those vertical dotted lines being when we applied treatments. And so what I really want you to take away from this graph is that we start to see these groupings emerge based on treatment. So here we have our soil only treatments, whether that was high sucrose or low sucrose, followed by our foliar only treatments, kind of following a similar grouping, and then our foliar plus soil treatments, yet again grouped here. So this to us is suggesting that treatment method really matters, but the amount that we feed those microbes maybe matters less, because we get these groupings regardless of whether it was a high or a low sucrose treatment for those. And also, contrary to what we thought might happen, the soil-only treatments actually here seemed to improve the ability for Phragmites to produce leaves, whereas our foliar treatments, similar impacts like we saw in prior years, really stark reduction in cover. And then our combination of foliar and soil treatments kind of falling right in there between. So it seems like strictly soil treatments are having maybe a neutral to stimulatory effect on this. But when we looked at our mortality data, we start to see a slightly different grouping in that anything that received a foliar application, whether that was solely foliar or the combination of foliar plus soil, had a pretty significant impact on mortality of stems. So it really seems like the foliar aspect of it is kind of driving impacts on the plant at this point, whereas our high soil treatments kind of fell right in line with our controls in terms of mortality of the above ground parts of the plant. And so this is, some data here to show, but visually this was also pretty apparent in the field. So here you can see on the left a control plant that is looking quite dead at this point. It's late in the season. This is late October now, so quite far into the senescence of this plant. Whereas our soil only treatments here, you can see same day that these were taken, or within a day these were taken, still quite green. Seems like maybe we delayed senescence a little bit with supplementing the rhizosphere with our product, whereas our soil plus foliar treatment or our foliar only treatment had pretty much similar impacts in terms of above ground effects of this plant. And so how did this then translate into what's going on with that extensive rhizome resource bank below? And so here we can see some sort of similar groupings like we saw before, some similar reductions in carbohydrates with things that received a foliar treatment, but maybe less so, and less impact with our soil-only treatment that we have. So our soil-only treatment carbohydrate rates here are sitting at about the same levels as we saw in our control treatments, whereas our ones that received foliar treatments here, whether that was high or low sucrose, whether that was foliar only or foliar plus soil, so our both treatments here on the right, we did see some of those above ground impacts translating to effects below ground through reductions in their resource storage. And so ultimately we want these resources to be depleted, or those rhizomes to be dead and decomposing for this to work. But we are seeing some of those effects of the above ground negative impacts translating to some of the below ground resource reserve there. And so just kind of summarizing all of this up in terms of our soil treatments here, when looking at just the foliar treatments, we saw similar impacts at this site, so different characteristics at this site than the other. So those kind of impacts seem to translate pretty well. We saw those same reductions in cover, increased mortality of the above ground parts, increased stress on the plant, as well as reductions in those below ground reserves. However, unlike what we had previously thought, the soil-only treatment, so getting our component directly in contact with the microbes and the rhizosphere and the rhizomes themselves, seemed to either perform similar as just a control treatment or even maybe mitigate some of the effects of that foliar treatment. So that's not exactly what we wanted to see, but we definitely learned some things from this to take forward. And it seems like we're still having more of this contact effect on our plants versus ultimately what our goal is just to get more of a systemic effect on this plant. So having, spraying one part of the plant, having that kind of transduce or move throughout the whole plant and really impact the plant as a whole. Because like I led off, the rhizomes and that below ground stuff is really kind of the keystone in managing this plant. So where does that leave us with the future of this bioherbicide product? We're still working to improve the efficiency and efficacy of this, but it's some promising results here, showing some potential for this to have significant impacts and be another tool that managers can use. And so we're working to adjust some of those concentrations that we've learned through our high and low sucrose concentration tests, as well as adjusting maybe the arginine concentrations and some of the other things. Still working to just refine the overall formulation, and we're starting to look at some other amino acids to add to this component that can help with that arginine ethylene kind of cycle, to just kind of improve the microbe's ability to produce this excess ethylene gas. Also looking at possibly introducing different essential oils as a different cuticle stripper that maybe plays nicer with some of the microbes and things like that. Also, some of these site level differences that I highlighted at North Hydro and also have occurred at our Crystal Waters site, but it wasn't able to touch on that just in the length of this talk. So probing a little bit more at like, why were we seeing more impact in certain conditions versus others could really help to answer some of these questions. And then ultimately working to improve some of the systemic effects of this bioherbicide treatment, having more impact with less effort, with being able to influence this microbial community and working to categorize the effects of this. So I wasn't able to share in this presentation some of the work that has been done by us and partners with this microbial effects, but we are starting to document some of the compositional changes in the microbial community that are happening pre and post treatment in the soil and the roots and the rhizomes and in the tissues. And that work's largely been done by Dr. James White, as well as some of Dr. Keith Clay's lab group in Tulane. And so we're starting to sift through that data and see what kind of changes are happening and functionally what that means and how we can really work on dialing the right levers to hone in those microbial effects, with the ultimate goal of developing a product that we can transfer the technology to more of a commercial setting that an industry can kind of scale up and reduce the cost and increase the effectiveness, and ultimately bring this to market for managers like many of you here today to be able to use and manage your lands and just add to your toolbox and give you more options for managing this really difficult plant. And so with that, I'd like to thank our many partners that have given us access to their land, that have helped us with the science, as well as many of the USGS employees, students, and interns that have worked tirelessly through the hot, humid days in Michigan to apply these treatments, count Phragmites stems, set up these experiments. And I'd like to thank you all for being here today to learn more about our work. And I think with that, we still have some time for some questions. So I think I'll turn it back to Teresa here to kind of moderate that session. So thank you all.
Yeah, thank you, Spenser. That was incredibly fascinating. I can't stop thinking about the 160 plots you guys had at one site and how long that must have taken to do like percent cover.
Effort, for sure. Quite the effort. And it wasn't just me, it was a lot of us out there doing it. And yeah, some long days, but we got it done and we got some good results to show from it. So
yeah. That's awesome. So yeah, like we have a few, we have about 10 minutes and we probably can go a bit over if people are interested for Q&A. So I already see some questions in the Q&A function that I will just start. Also, that is Spenser's email on the screen. If you have a question now, but you don't know how to articulate it or want to reach out to him after I asked permission, he said he's more than welcome. So
absolutely.
Okay, so first question from Jill. Have there been any tests of treating an entire patch? Could plants within the small plot might be getting resources from the untreated plants outside the plot? and therefore reducing the impact of the treatment?
Yeah, great question, Jill. Thank you for that. Yeah, that's definitely something that we've talked about, is treating kind of, transitioning from these smaller patches to maybe a whole patch approach so we don't get the influence of sharing resources. Like I said, we do the best we can with severing the connections to our experimental plots, but I'm sure most of you are aware those rhizomes, quite dense, can go quite deep. The majority do occur within the first 18 inches or so of the soil typically. And so, yeah, that could be part of our next steps, is looking at finding, some of those patches we work in are quite large. So treating these larger patches would be difficult, but there definitely is some potential for us to look at treating whole patches with this product to look at impacts, reducing, like, as you say, the ability for them to share resources across across the existing patch to our treated patches. So something to think about and move forward this coming summer and in the future of this testing for sure. So thank you, Jill.
Yeah, that's a great thought. And this question's from Megan. Would you expect to see the late foliar treatment to impact the plants the following season more, or is the intent to see the greater impact at the end of the season?
Yeah, that's an interesting question about terms of the timing, and is the late going to impact it more versus the early? I would say that the impacts are definitely most noticeable right at the end of the season, after all of those treatments have taken effect. And our early treatments and our late treatments do occur essentially at the same time. So our early treatments start in June, but they occur every 4 weeks. And then when we get to what would be our third treatment that season, that's when we start our late, but we're also still applying early. And so I think it's a little bit hard to see at the beginning of the season, like, regrowth between early and late, because they're kind of still getting the same treatment applications. But that was our thinking with doing the late treatment, was, reducing the labor. One is how many times we have to apply this product, and also hopefully having more holdover to the next season as we're really hammering the plants at the end of the growing season before they're able to translocate all of those resources and such to the below ground parts of the plant. So interesting question.
Next one, from Ray. What % carbohydrate reduction, I guess, do you think would be necessary to actually achieve quote unquote control of the species?
Yeah, that's a good question. It's a tough question. I mean,
Yeah.
we've been working with the carbohydrate data, in not just in this, but some other work that we did with some cut to drowned, or cutting the plants underwater experiments, and looking at how that impacts carbohydrates. And it's hard to say. There's a fair amount of research out there in terms of carbohydrate resource dynamics of plants. And there's a lot of interactions that happen in the soil in terms of whether it's an anaerobic versus anaerobic process. Anaerobic soils, or it costs more energy to break down carbohydrates and use them. So probably in those situations, the resource reserves might not need to be drawn down as much, versus soil characteristics that operate more on aerobic processes probably would need to draw down those carbohydrate reserves even more. But that's definitely something that we've seen throughout the years, is there's a lot of seasonality, not just with our treatments, but just in terms of the growth of the plants and how much resources they're able to store. So I don't have necessarily like a number. 5% of carbohydrates is the magic number, right? Like, I think it's dependent on the site and dependent on the soil characteristics and the properties and things that are occurring within those rhizomes themselves, because we're seeing reductions down 5% of those plants, and that still seems to be enough to support at least initial regrowth the next season, where they can start photosynthesizing and replenishing those microbes. But one thing that we've turned to the literature and our scheming, how to maybe look at how quickly are things actually moving in the plant, like how quickly are the resources that are produced through photosynthesis like actually moved down into the roots. Like, are we spraying our thing and it's signaling the plant stress and it's just like saying, uh-oh, we got to take in our resources, or is it kind of more of a slow drip kind of thing? And so those are things that we're trying to think about, how we can design some experiments to look at like the rate of carbohydrate movement in these plants.
The research never ends.
Yeah.
Is there any evidence that the rate of tiller growth is impacted by any of the treatments that y'all applied?
Yeah. So it's in our field experiments that we do, we don't excavate the whole plot really to look at, total biomass down there or how things are growing, because we tend to go back to them future years. And so we try to minimize disturbance and we take rhizome samples the best we can. But in terms of tiller growth through some greenhouse and some laboratory studies that we can actually harvest the whole plant and look at that. We are seeing impacts on like the health of the roots and the health of the rhizomes in these greenhouse and laboratory settings, and how much they're able to grow during a season, and like how many root hairs are on the plant, which affects their ability to uptake nutrients and water and things like that. But we haven't directly like looked at the rate of elongation or the rate of growth of rhizome tissues below ground. But that's something that could be interesting to look at for sure.
Yeah. And another question, probably a lot of people also thinking about it. Did you guys notice any non-target effects from the treatment? Did you notice plants growing in the experimental plots after the treatment were applied?
Yeah, for sure. So it does not seem like our product at this point is acting species specific to Phragmites.
Sure.
It's more of a broad impact. So we do, if it comes in contact with other plants, it does have similar effects that we see with Phragmites, but those effects are limited in time. We see a lot of regrowth within our plots within the season as well as between seasons. And you could kind of see that in some of the images I showed at the postseason. Some of our treatments had a pretty good understory of plants and things being recruited. And so these components that we use are all biodegradable and or photodegrade within like 30 days in the environment typically. So things like Roundup and imazapyr, glyphosate and imazapyr have been shown to have a little bit longer impacts in the soil and things like that. And so far, our products been shown to have pretty limited time frame impact and have future recruitment of other plants that occur in the area for sure.
Yeah. Another question about how frag specific is this work? I think you kind of just addressed it, but if there's anything else you want to add on to that.
Yeah, no, I mean, I think that covers it. I mean, application of it, we try to just target the Phragmites as much as we can. We use what we call a spray to drip method. So we essentially just spray the plant until it's fully saturated and starting to drip off of the tissues like initially. And then that's kind of when we stop our application. So we limit our impact on other plants that way. But like I said, we are seeing kind of more of a broad impact across different types of plants. And that's something we thought about looking at is, does it impact forbs versus grasses more, woody plants? And so those are some things that future of this work to look into is what range of plants is this having an impact on?
Yeah, it seems like there's so much to do with this question that the questions are endless. Okay, a few questions from Christopher. So I'm just going to start with the first one. It seems this process seems very labor intensive, even with the every 4 weeks. Is there, I'm guessing an estimate, you probably don't know off the top of your head, but what is the cost versus this cost versus an herbicide treatment cost? And like, how equivalent are they?
Yeah, sure. So currently, like, the 4-week treatment is fairly labor intensive and typically more than managers do or have the ability to do. And so, these are some initial tests that we're doing on this and still working to improve the efficiency and improve the formulation and really dial in what the optimal formulation is with that. And so I don't have like a direct cost comparison of our product versus something like glyphosate or imazapyr that might be used, because application rates are going to be variable, but depending on the density of the stand, age of the stand, all those kind of things to compare. But we are working towards developing a product that is going to be more, as efficient or more efficient and less as labor intensive or more so, or I guess less labor intensive and costly than some of these traditional approaches. So we don't envision this as being like the silver bullet to end all other treatments and here's our approach. It's more of just giving more options for managers to be able to use, and particularly in areas where traditional herbicides might not be welcome or might not even be permitted to be used, just additional tools for managers to use that are as good or better than the existing ones.
Good answer. And then I hope I'm pronouncing this correctly. Is it arginine? Is that one of the components of the cocktail?
Yeah, it's one of those words. Arginine, okay. Arginine.
Arginine.
Tomato.
That arginine, is it easy to get? And if so, how much does it cost?
Yeah, it's pretty easy to get. Obviously, the more bulk, the larger quantities you buy, the cheaper it is. We buy it in not huge amounts because we don't want to just waste a bunch of resources and buy excessive amounts. So we kind of purchase it throughout the season based on how much we're applying, and things like that. And it is the one we buy, it's a human health supplement. So people actually consume it for nutritional purposes and things like that. So it's totally safe in terms of that for humans and such. So it's readily available through a number of different outlets to acquire and things like that. So
Interesting.
You don't need a special permit or anything like that. You can just go buy it off the shelf pretty much.
Okay, a question from Ray. Have the previously treated plots been revisited to assess the following season's regrowth?
Yes. So in all of the sites that I showed today, there's a couple others that we've been to that just the sake of a 40-minute webinar I couldn't get to. All of the plots we have revisited the next year and really applied subsequent treatments the next year, but we do initially monitor those plots at the beginning of the season for signs of regrowth before we start applying treatments. And so there's, maybe some slight impacts on regrowth the following year, but it's not strong, visual impacts that we're seeing or in terms of our monitoring data and things like that. So that's something that we're definitely working towards looking at, improving the systemic nature of this as a way to have these longer term holdover effects from year to year as we move towards kind of a product that can be used by not just us, but kind of more wide scale use for sure.
Yep. Okay. Nicole, am I missing any? It seems like I've gone through the list, but.
Yep, I don't see any on my end.
Okay. Now is your last chance. If you had a question, throw it in the chat so I can read it.
If you think of one later,
Okay.
There's my email, so feel free to reach out. And happy to.
Well, I was going to say thank you. That was so interesting, and I'm sure we're going to need another, maybe like in a few years, we definitely need another update on this work because this is so fascinating. Oh, wait, I do think I see one might have popped up.
Sure.
Oh, this is just an excellent presentation.
Thank you. Thank you.
And then just for you guys, if you wanted to share this presentation, if you wanted to come back to it, it is recorded. You'll get an email that says like view the webinar recording, and it'll also be on like our YouTube and website. So feel free to share with colleagues if you know people who are interested and want to hear this. And then just a shameless plug that we're always looking for new science to promote. So if you on this call or anyone colleagues that you know of that are working with Phragmites, it doesn't have to be in the Great Lakes region, and want to present at our webinar, just email us and we'd be happy to have you. So thank you for watching, and thank you, Spenser. Thank you, USGS. And great webinar. Thank you for your time.
Yes. Thank you all for being here.
Mm-hmm.
Bye.
Thank you.